Good morning. We are going to do transport across biological membrane, the very first competency 1.1, the very first. And we are going to do passive transport today. There are various ways in which transport can occur. It can be passive transport, which is going to be topic of this lecture. It occurs along a concentration gradient and there is no energy expenditure. Since it is occurring along a concentration gradient from high concentration to low concentration, so therefore there is no energy, metabolic energy is not used. Later, we are going to do active transport where energy would be used and vesicular transport, which is for the larger macromolecules. But at the moment, let us concentrate on the topic passive transport. Now, passive transport can be unaided when it is called simple diffusion, or it is aided by transport protein, in which case it is called facilitated transport. So simple diffusion along a concentration gradient, unaided. So it simply, the solute moves from high concentration to low concentration. So evidently, the biological membrane should be permeable. Only those substances can undergo simple diffusion to which the biological membrane is permeable. What are those substances? The substances are those. We must actually have an outline of the membrane structure before we come to those substances. The membrane is only 2 molecule thick. This elegantly simple design is common to all organisms. It is referred to as lipid bilayer, which is predominantly hydrophobic. As you can see here, only 2 molecules, lipid bilayer, and predominantly it is hydrophobic. Mostly, they are phospholipids, which have got a small hydrophilic portion here, this one, and the long hydrophobic tail, which is this one, long. So therefore, it has got a predominant hydrophobic character. So only those substances are able to pass through it which are lipid soluble and which are small molecules. These two criteria must be met because the membrane is predominantly hydrophobic. So only the hydrophobic non-polar lipid soluble substances of small size are able to pass through it unaided. Few exceptions also we are going to see. Now, some such substances which can pass unhindered spontaneously are blood gases. These are not only small but non-polar also. Small lipid-soluble molecules like steroids, fatty acids, and very small polar molecule. Polar molecule only if they are very small. For example, water itself, urea, glycerol. These substances can pass unhindered. Very small polar molecule restricted as shown by the dotted line. And the water-soluble substances, they are rejected by the hydrophobic core. They cannot cross. Not only these, large hydrophobic, for example, triglyceride, large lipids, they are also rejected by the hydrophobic core. So just to summarize, the simple diffusion is limited to small lipid-soluble molecules as shown here. So they pass through this much of hydrophobic core easily. So simple diffusion, just to repeat, downhill, no energy expenditure, no carrier is required. Now we are going to do facilitated, which are same. Concentration energy expenditure का same है, but they do require a carrier And what is the carrier? Carrier are membrane proteins. So facilitated transport movement occurs with the help of transport proteins called translocases. And many water-soluble nutrients which we saw cannot pass spontaneously through the membrane. ये translocases की मदद से they can pass through the membrane. So the translocases are either channels or there are carrier proteins. We are going to see them one by one. First, carrier proteins. Carrier proteins pass through a transport cycle during which there is conformation changes. It oscillates between two conformational states we are going to see, and that is appropriately called ping pong reaction. There are four steps: binding of solute to the transporter, and the transporter undergoes conformation change, as we can see here. First, we are considering glucose transporter, binding of the glucose to the transporter, as a result of which, this is first step, as a result of which there is conformation change. You can compare these two structures, two conformations. This is called outside open conformation, and this is inside open conformation. Now, third step is release of the solute. So that is glucose released into the cytosol. And the fourth step is again a conformation change. Here you can see again a conformation change to the outside open. So by oscillating between two conformation states, glucose has been transported into the cytosol. So it occurs along the concentration gradient. Now one may ask, glucose, the way it has entered, can it not move out in the same way? No, it cannot. No, it cannot. Because intracellular utilization of glucose ensures that the concentration gradient is maintained. Now, four important features of carrier-mediated transport. One, very specific for the substrate. For example, जो glucose transporter हमने देखा था, it can transport D glucose but not L glucose. Not even the mirror image it can transport. Second, saturation kinetics. It shows a hyperbolic curve. Why? Because rate of transport is limited by number of the carrier proteins, and these proteins get saturated as the concentration of the substrate rises. So you can see here, as the substrate concentration rises, the velocity shows a fall. This is in sharp contrast to the simple diffusion where it's a linear curve. Third, specific inhibition. For example, glucose transporter can be inhibited by glutor. So a number of drugs of this type are, it's a glucose analog, a number of more drugs are being developed. Then there is physiological regulation. The carriers that bring glucose into adipose and muscles, for example, are activated by insulin. We will do more about them. These carriers, they are activated, upregulated actually. Likewise, ADH ka aquaporin we are going to do in today's class only. Then we come to, after this, saturation kinetics. I think we have finished most of the things about it. So carrier protein ko humne finish kar liya. Now we are coming to ion channels. There are certain transmembrane proteins which traverse whole length of the lipid bilayer, and a pore exists in middle of some of these. So they make a pore of about 5 to 8 nanometer, and they transport specific ions like sodium, potassium, calcium, and they are oligomeric and they are very specific. For example, सोडियम ट्रांसपोर्टर सिर्फ सोडियम करेगा, इट विल नॉट टच potassium and vice versa. And these channels are regulated. Opening and closing of these channels are regulated by neurotransmitters or by membrane potential. Accordingly, three types, I will very quickly go through them: voltage gated, ligand gated channel, and mechanically gated channel. Voltage gated channel, they respond to change in the electric potential. They open or close based on the change in the electric potential. For example, sodium potassium channel, you know about their role in the nerve conduction. Sodium channels, when they open, depolarization occur. Potassium का जब होता है movement, then repolarization. Ligand gated, they respond to hormones, metabolites sometimes, or drugs. For example, acetylcholine receptor. Acetylcholine receptor, when acetylcholine bind to these receptor, this results in opening of the sodium channel and regeneration of some potential, etc. You will learn more in physiology. Mechanically gated, they respond to touch, vibration, pressure, etc. So we have done most of the things about ion channel, carrier mediated. Now just one or two clinical correlates. Diabetes insipidus, it's a condition where ADH mediated reabsorption of water is hampered. There are certain transporter, actually ion channels, in the collecting duct of the renal nephron collecting duct level per, these aquaporins are present. And these are pores, actually, as the name pores for water, aquaporin. They facilitate movement of water across the tubular membrane. In other words, renal reabsorption of water. So mutation in this aquaporin results in less reabsorption of water, so very dilute urine in large quantity is produced. So this is actually aquaporin is under control of a hormone called posterior pituitary hormone, antidiuretic hormone. Second condition, cystic fibrosis. There is a protein called CFTR, cystic fibrosis transmembrane conductance regulator. It is actually an ion channel through which chloride passes, and when chloride passes through it, it is accompanied by water. So mutation in this protein, water का movement भी hampered हो गई. As a result, the secretions become thick. For example, pancreas के अंदर, if the CFTR is mutated, then what happens? The pancreatic secretion will get thick, they will get viscous, resulting ultimately in plugging of the small ducts. As a result, more stagnation, pancreatitis will occur. Lungs, by the same way, repeated lung infections will be there. Sweat glands, chloride will deposit on the skin, in fact, sodium chloride, and the parents, they complain of a salty kiss. Whenever they affectionately kiss their child, salt ka taste aata hai. So you can extrapolate why that happens. So overview, concentration gradient, yes. Energy expenditure, no. यहाँ पर भी unaided, and this is aided by either by ion channels or by carrier mediated protein. So finally, key point, spontaneous diffusion, simple diffusion में downhill, and rate is solely determined by concentration gradient. So this facilitated is occurring with the help of integral membrane protein, which facilitate movement of solute molecules along a concentration gradient. So a number of water-soluble metabolites and the other excretory products, etc., which cannot pass by simple diffusion, their movement is made possible by facilitated. But common to them, no energy expenditure occurs, no metabolic energy is expended. So active we will do later on. Here, expenditure of energy and against a concentration gradient, we will see in the next class. Thank you so much.